Hello, I am one of the Year 13 students who is involved in the Authentic Biology project at Coltham School. We are a Bristol-based school who have been working in the partnership with the University of Bristol. Over the last 2 years, we have been studying the expression of genes we believe to be associated with the early onset of osteoarthritis. With the support of the Bristol University academics, and in particular the final year biochemistry undergraduates, we've learnt how to search for these genes, design primers for them, and subsequently use PCR and gel electrophoresis to further investigate when they are expressed during the early stages of embryonic development. The undergraduates have opted to do a final year school-based dissertation, and I think that I can say on behalf of all of the authentic biologists that we have benefited massively from their involvement. We use zebrafish as our animal models to study gene expression due to a number of reasons. For example, they can reproduce rapidly and are very easy to maintain. But most importantly, zebrafish embryos are large enough to visualize and manipulate under the microscope, and their transparency during early development would allow us to visualize labeled genes in situ. May will now go on to discuss how our osteoarthritis genes are selected.
To attain a list of genes that might be involved, the University of Bristol used genome-wide association studies. GWAS are relatively new ways to identify certain genes that may be involved with human diseases. They are conducted by taking DNA samples, usually by blood samples or cheek swabs, from two sets of people, those with the disease and those without. Their genomes are then scanned for small variations called single nucleotide polymorphisms, or SNPs for short. If certain genetic variations are found to be significantly more frequent in people with the disease compared to people without the disease, these variations are then said to be associated with the disease. These variations can then help pinpoint the region on the genome where the disease-causing problem resides. Each case looks at many thousands of SNPs in thousands of people's genomes. To generate this sort of data, large numbers of cases and controls are genotyped. This level of analysis can be done by cohort studies, such as the Bristol-based, even longitudinal study of parents and children, often called the Children of the 90s, or just ALSPAC, and is a world-leading birth cohort study. It was set up in 1991 and recruited 14,000 pregnant women. These women, their children, and sometimes their children's children, have been studied, and over the last 25 years, a rich resource of phenotypic, environmental, genetic, and linkage data has been provided. 3 GWAS studies carried out by the ARCOGEN Consortium, which is an osteoarthritis study funded by Arthritis UK, which used samples from ALSPAC. SNPs associated with osteoarthritis have been found using Manhattan plots. They're named after their resemblance to the Manhattan skyline. Each dot represents a SNP with their chromosome position along the x-axis, and each of the blocks of colour representing a different chromosome, and their association on the y-axis. If any SNPs are above the line of significance, which is the red line that goes across, the ones labelled with the red ring, they are then said to be associated. Through the ARCOGEN GWAS study, the university found 9 genes for us to investigate. Nick will now go on to explain our technique and results for these 9 genes.
Right, okay. Because we're looking at the expression of these genes in embryonic zebrafish, we have to obtain their RNA. The RNA is extracted from zebrafish embryos and provided by the university to us. So to the RNA, from days 1 to 6 post-fertilization, we then add reagents from a cDNA synthesis kit to make our cDNA templates. These should only contain genes that are expressed on that particular day. So remember, we have 9 genes to research, so for each gene, we need to look at its expression each day post-fertilization. Using STAB1 as an example, we add the forward and reverse primer for this gene, plus our cDNA template and other necessary reagents like master mix to a small 0.2 millimeter Eppendorf tube, which is then placed in the thermocycler. If the gene is expressed, then the primers will find their complementary sequence on the template and the gene in question will be replicated many times and is amplified. After PCR, we use gel electrophoresis to visualize on which days post-fertilization the genes are expressed. The genes are then stained and the gels from this are then stained and photographed under UV light. On our pictures, the bands are quite faint, as over the year we have sort of experimented with different stains to try and get the best results, and some have been more successful than others. Thanks to all the Wellcome Trust funding, a lot of the equipment we need to carry out the investigation is available for us to use in the science department, and we are often able to use work in free periods and in small groups without teacher supervision. And in some cases, we've been down to the university to stain the gels with ethidium bromide, which is one of the gels that we can't use at school. As you can see from the table, the genes STAB1, PBRM1, MYA6 and SENP6 were expressed on all 6 days post-fertilisation, indicating they are involved in the early development of zebrafish and, by extension, may contribute to this early onset of osteoarthritis. GNL3, while not expressed every day, was still expressed. The others, however, weren't expressed on our gels. MUSTM1A has been a bit of an anomaly to us. Either it was expressed on all days, but due to several faint bands being visible on each day, we have come to the conclusion that this could be non-specific binding. This is when primal molecules attach to random regions of DNA, producing strands of varying size. To continue this research, we would then carry out in situ hybridization or immunohistochemistry to label the genes and see where exactly they're expressed in the zebrafish. Eden is now going to talk about the direction in which we're taking the project this year.
So I am Coltham School's Year 12 representative. I joined the project at the start of the academic year, and the new focus this year is to look at another disease other than osteoarthritis. This year, we began looking at genes that are associated with heart failure. Again, similar to our osteoarthritis research, we use zebrafish as models for humans, as they have a regenerating system. After a human gets a heart attack, a scar tissue is formed around the damaged area of the heart. Scar tissues are mostly made out of collagen, which is a tough, fibristic protein. However, for a zebrafish, they can completely reform their heart. A scar does form, but eventually it goes away. By mimicking heart attacks onto the zebrafish, we can monitor what proteins are present and what processes occur during the regeneration of the tissue. Again, similar to the early stages of our osteoarthritis research, we again looked at genes associated with heart failure using GWAS. Using GWAS, we searched up the genes that were found in heart failure. We tried to narrow down the genes depending on the amount of associations it had, which had to be below 20. We then went on to the ZFIN database to see if they had zebrafish orthologs. If they did, we would conduct further research onto the genes we found. We then picked the top 6 genes, which were the most interesting, and here they are. For example, the reason why we picked the gene EVX1 is because it has a regenerating property, and this, we believe, would be very useful for our research. For the rest of the academic year, we will look at genes expressed in the hearts of zebrafish. This will feed into the research led by Dr. Beck Richardson at Bristol University. If you would like to see where our research leads this year, follow our Instagram. Thank you very much for listening. Thank you. Any questions?
Hi, that was a fantastic talk. Really enjoyed that. Obviously, we're from Tapton, so we're also working on zebrafish, and we're looking at heart disease. And one of your genes that you just put up there, TBC1D4, do you have any idea what it does? Because that came up in one of our screens, but we're not looking at it at present.
Somewhere there'll be something. Is it back on that slide? We do have a document. I don't think we have it on us about the different genes because we just did this the other week.
There's different versions, isn't there? The one that came up in our human screen was TBC1D19. Maybe we should chat about it at the poster.
Yeah.
And sounds like a plan, doesn't it? Awesome.
Not be treated with anyone's tongue, shall So good. Another question, please. Can I ask, when you showed you had obviously you can't use ethyl bromide in schools, but you're using slide green, yeah? Do you know the level of sensitivity compared to ethyl bromide? Is it like 1,000-fold or? I'm just curious.
Well, the ethyl bromide, it's photographed in the machine, so it's a lot clearer. It can be it's a higher quality than we can use.
Sure. Question, yeah?
Hi, yeah, fantastic presentation. My question actually applies probably to all of the participants today, but I wanted to get a sense of how your perceptions of science have changed from how they were when you were, say, in year 11, from having been involved in authentic biology, and has it changed your aspirations in terms of what you want to study going forward, or what sort of career you might be adopting further down the line?
One at a time, there you go.
I suppose I'd never really heard of biochemistry beforehand, and we have wonderful undergraduate third years, like who's one over there, and they're just amazing at helping with talking about what they're doing in their school and about degrees and applying. And I've now applied to do biochemistry, so without this I would never have thought of that and doing the whole genetic things. And yeah.
Anybody else? Yeah.
I've personally have learnt a lot about the scientific method and how obviously not everything goes as you want it to, and you do have to keep working on refining your technique. So it may not be about the results you get, but like working through the process and doing it sort of again and trying to refine it is pretty rewarding.
Too early or?
Well, clearly, I just recently joined. At the start, I actually didn't know what I was doing with authentic biology because I did not know what it was. But then when I came, the undergraduates clearly helped me with everything, and I understood why zebrafish were used, because previously I didn't even understand how fish would relate to anything. But then I understood that they have a regenerating property, and it could actually help, and they could just keep on using the same the zebrafish species, and it's actually definitely changed my perspective for it.
Great. Okay, right. Okay, good question. Thank you. Any other questions? We've got a bit of time, so I'll keep them up here. So, yep. No. Yeah, well, I think one of the strengths that I've picked up from the Coltham interaction is that you're having a lot of undergraduates coming in. Is it a 6-week placement in 3 blocks, or how does it work?
So we're working over about 10 weeks. We do one main session a week on Thursday, and that's after school. So all the work they're doing, they're coming in afterwards, and we're working with them. And then we've been guiding them with creating their presentation, but this was work that was done with the last year's undergraduates, so it's more of just helping them refine. They did all of this work by themselves, and they've been fantastic. Hope we've had some really good results so far this year, and hopefully it will continue that way.
Really strong support from the university, from the university. And you get credits for this? Is it part of your course?
So it's going to form part of our final year project. So we will be both reviewing the techniques that we've used in the lab, but also reviewing our teaching methods and delivery.
Excellent. Thank you. Any other questions? Okay, you're off the hook.